Regulation of gene expression in prokaryotes. Prokaryotes have operons which are functionally related genes grouped together on the chromosomes. These group of genes, which are called as operons, they are switched on and off together or simultaneously with the help of a single promoter. There are specific mechanisms that involve the rapid turn on and turn off of gene expression in response to the environmental changes. Prokaryotes have pre-programmed circuits or cascades or systems or pathways of gene expression that consist of proper sequence of the events. Here we can see that how there is a regulation of gene expression in case of prokaryotes. They usually have genes for the enzymes. That's the example of, in case of tryptophan synthesis, genes for enzymes that synthesize the amino acid tryptophan. There are separate regulatory genes that makes the repressor protein that actually control the expression of the genes in case of tryptophan synthesis. Repressor is activated by binding tryptophan and blocks the transcription by binding operator. Negative feedback shut down the operon if there is plenty of tryptophan present. So these are some of the cascades or systems that how gene expression is regulated in case of prokaryotes. In this diagram, we try to understand that regulation of gene expression in prokaryotes. There is a separate gene that produces the messenger RNA that is actually the regulatory protein. So here is the tryptophan that we can genes of the operon, that is A, B, C, D, E. Here is the operator and this is the promoter region. So if the tryptophan is absent, so the repressor inactive. So when repressor is inactive, so it will not bind to the operator region. So the operon is on. So it will produce the polypeptides that make up the enzyme for the tryptophan synthesis. Here we can also see that DNA, there is a separate gene that produces the messenger RNA that is actually producing a protein that is a regulatory protein. If there is a tryptophan co-repressor, due to this tryptophan co-repressor, the repressor becomes active. When it becomes active repressor, then it will no more bind to the operator region and the operon, it becomes off. Another example, tryptophan present, repressor is active and the operon is on. If it is active, then there is no production or synthesis of the RNA molecule. If we conclude that what is the regulation of gene expression in case of prokaryotes, prokaryotes have operons which are functionally related genes which are grouped together on the chromosome. This group of genes, which is called as operons, they switch on and they switch off together.